This is Lecture 2 for Biology 177, Anatomy and Physiology. Today's topic will be body cavities and chemistry. Body cavities are actually formed during the development for humans. Humans are vertebrates. This means they're animals with a vertebral column that protects and houses the spinal cord. Now, during early development, we actually form 3 tissue layers. You'll notice the diagram here at the bottom starts with us as a zygote, a complete cell. From there, we actually divide. We then form cleavage furrows as we divide into an 8-cell stage and then into a blastula. A blastula is a hollow ball of cells. The inner area is going to be a cavity called a blastocoel. The blastocoel is actually going to then start to divide and grow into other overall layers. These layers, as noted on your left-hand side, are the ectoderm, mesoderm, and endoderm. The ectoderm is going to be the outermost layer, highlighted in blue on the diagram. It forms the skin and nervous tissue later on during development. The mesoderm is actually going to be inner portions of the endoderm and is going to form the organs and muscles of the body. The endoderm, or the innermost lining, is actually going to be your digestive tract or alimentary canal. Here we can actually see where the blastula is going to fold inward into a gastrulation. These gastrulations are going to create fluid-filled cavities called the coleums, and they will actually form into the different areas of the ectoderm on the outside, the endoderm on the very inside, and the mesoderm identified in blue here. From there, we'll actually open to where we kind of form a human donut, to where we have a mouth on one end and the anus on the other, with the central portion being the digestive tube or alimentary canal. Here we actually have a visual representation of the alimentary canal. The alimentary canal or digestive tract runs from the mouth or oral cavity all the way to the anus. The lumen or opening, the whole opening that runs through the entire central portion of the alimentary canal, is actually part of the external environment. This means that we have a hole running through the center of our body that's separated by different sphincters. These sphincters allow us to control the flow of different food or liquid items as it passes through the digestive tract. So remember that this is, again, continuous with the external environment. So we are a human donut, effectively. Now, the different body cavities house many organs within that interior area, and these body cavities are also known as coleums. The body cavities of the trunk contain organs of the respiratory, cardiovascular, digestive, urinary, and reproductive systems, and they're going to be divided into different areas. Now, true body cavities are actually lined by serous membranes. Serous membranes are designed to protect delicate organs and permit movement or changes in size or shape of the organ. So they also help to protect against friction or any sort of rubbing that may occur. Here we can actually see the 3 serous cavities. They're the pericardial, pleural, and peritoneal serous cavities. Notice on the right-hand side where you see the fist pushing into a fluid-filled balloon. This is an analogy for how the heart and pericardial cavity have their relationship with one another. In this example, the fist represents the heart. Now, the actual inner lining of that balloon is going to represent what we call the visceral layer of the serous membrane. Visceral means organ or touching the organ. The outer portion that is not touching the fist is actually going to be called the parietal serous membrane. Parietal means away from or exposed to an outer surface. In this case, it is a fluid-filled membrane, and this is again designed to act as almost like an airbag, protecting against friction so the organ can move without rubbing or causing abrasion to it. Now, the pericardial is cardial, heart. It surrounds the heart. This is lubricated and allows the heart to beat. So that's the first of the serous membranes, the pericardial. The next is the pleural. The pleural is going to surround the lungs, and this is going to permit movement during breathing. The final is the peritoneal. This is going to actually house the digestive organs as well as some accessory organs. The peritoneum can further be divided into the retroperitoneal. This is a continuation of the peritoneum that is going to house the kidneys and the pancreas. So to simplify, pericardial surrounds the heart, pleural surrounds the lungs, peritoneal surrounds the digestive tract and some reproductive organs. Here we can see a better view of the actual pleural cavity and the different coverings that go around the heart as well. So the pleural cavity, again, surrounding the lungs, we can see the visceral and parietal pleura. And then on the heart, we can also see the breakdown of the pericardium and the different layers that surround that. Here we can actually see the peritoneum to where it's going to cover the entire digestive tract from the liver, the stomach, the small and large intestine, and then the retroperitoneum in the back part of the actual abdominal cavity that covers the pancreas and the kidneys. So this peritoneum is going to be combined with the others to form the three serous membranes of the body. I'll remind you one more time. Pericardial is going to be heart, pleural is lungs, peritoneum is digestive tract and reproductive organs. These are the three serous membranes inside the body cavities. Switching gears a bit, we're now going to start focusing on chemistry basics, as a lot of these understanding of the bonds are going to affect us later on when dealing with a lot of physiological processes. Here we can actually see the overall basics and layout of an atom. An atom is going to make up the different elements of the universe. Atoms are composed of protons and electrons, and in some cases, a lot of times, neutrons as well. We can see in the upper left-hand corner, hydrogen is going to be one proton and one electron. Where the proton is going to be the nucleus of the atom. Just to the right of hydrogen, we have carbon. Carbon is going to be atomic number 6. This is because it has 6 protons in it, and this is going to also determine the number of electrons that it has. Protons and electrons are always the same with every single element. If it has 20 protons, it will have 20 electrons. They balance each other out. Protons have a positive charge. Electrons have a negative charge. Neutrons, which are sometimes found in the nucleus, are going to have a neutral charge. Neutrons typically are going to have the same amount as protons, but in some cases, it doesn't. In the case where we actually don't have the same number of neutrons as protons in the nucleus, we call this an isotope. An isotope is where we have more neutrons than normal. In a lot of cases, this causes a decay, and this decay is going to produce beta particles as the neutrons break off. This is what we know of as radiation. That's where you hear isotopes always being referred to in the case of radioactive isotopes, specifically uranium, or in this case, something like carbon, carbon-13 or 14, which is going to have more neutrons than protons. The next thing to note within chemistry is going to be chemical bonds. Now, you have an individual atom that's composed of protons, neutrons, and electrons. Electrons surround the overall protons and neutrons, the nucleus. The electrons are going to be in a cloud around the nucleus. And when two atoms meet up, they might actually bind together. They bind via the electron cloud, and in a lot of cases, they share electrons. This sharing of electrons is known as a bond, and specifically a covalent bond. Now, there are different types of covalent bonds that we actually have. There's nonpolar and polar covalent bonds. Nonpolar covalent bonds, the bonding of electrons shared is equal between the two atoms. So there's no charges on the atoms, and it's balanced all the way through. There's no polar ends. However, an example here, as you can see with item B, is going to be hydrogen chloride. This is a polar covalent bond. In a polar covalent bond, the bonding between electrons shared is unequal between the two atoms. So there are partial charges on both atoms. In this case, hydrogen is going to have a positive end, whereas chloride has the negative end in this case. So it's going to create a different charge on one end. Covalent bonds are shared electrons. That's the key thing to remember here. Covalent equals shared. They share the electrons equally. In some cases, there is a complete transfer of bonds, or a transfer of an electron. This transfer of an electron is called an ionic bond. An ionic bond is the complete transfer of one or more valence electrons. The full charge is on the resulting ion. Great example of this is sodium chloride, more commonly known as table salt. Another type of non-covalent bond involves specifically molecules of water. Here we can actually see the oxygen molecule with two hydrogen molecules. We know this is H2O, it's water. Now, a molecule of water in itself is a polar covalent bond. We can see it has a positive hydrogen, a negative oxygen, and they're bound together. Now, when you have multiple molecules of water together, they actually have a attraction between them, between the hydrogen and between the oxygen. It is a weak bond, and that weak bond is called a hydrogen bond. It is a unique bond to water molecules. Now, hydrogen bonds themselves are weak individually, but the huge number of them in living organisms makes the bonds extremely important. So in this case, we actually can develop the surface tension of water with these hydrogen bonds, and it is what makes water the universal solvent, because it is a loose bond that can actually divide and combine with other things. So this is why it's so easy to dissolve sugar or salt or a variety of other compounds directly within water. Now, another concept to understand with respect to the hydrogen bonds is looking at different interactions with water. This is looking at hydrophobic versus hydrophilic. Phobic is a key word that we remember. Phobic means fear. Hydrophobic is water hating. It occurs between molecules which cannot both participate in hydrogen bonding, so it doesn't like water. A great example of this is going to be any sort of oil compound. Any sort of lipid or oil does not like to interact with water. It divides with it and can't bind with it readily. Hydrophilic means water loving. So hydrophilic interactions are going to occur between molecules that can participate in hydrogen bonding. Things like sodium chloride, it's going to be glucose, sucrose, sugar, things like that. They will bind very readily and dissolve very readily in water as well. This is going to be a huge thing when we actually look at cell membranes and how cell membranes control the flow of things into and out of them. And they do so with hydrophobic and hydrophilic interactions. Now, talking about common organic compounds in the body, now that we understand some of the basics of chemistry, we're looking at things that we might be comfortable from a dietary perspective. We're looking at carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates themselves are going to be your sugars. Sugars are monosaccharides, disaccharides, and polysaccharides. They are getting progressively more complicated as we add more chains to them. Carbohydrates serve the general function of energy supply, signaling, and structure in the body. Lipids are otherwise known as fats. Fats are made up of fatty acids. They are hydrophobic, water-hating. They are used for structural integrity, primarily in membranes. They are used for energy, and they're also used to manufacture hormones as well. Proteins are made up of amino acid chains, and they combine together to create more complex structures. They are used for structural components. They're used for signaling. They're used to transport things into and out of the cell or across the body, as well as a host of other functions that can eventually make down different enzymes or things like that that might catalyze different reactions. Nucleic acids are made up of nucleotides. Now, these are an amino acid base that's going to be converted down a little bit more simply, but it's effectively talking about genetic material in the case of DNA or deoxyribonucleic acid, and then signaling RNA. RNA is going to be the signaling component that will build DNA later. Now, of the overall 4 classes of organic compounds in our body, of carbohydrates, lipids, proteins, and nucleic acids, 3 of them, proteins, carbs, and nucleic acids, are macromolecules. Macromolecules are polymers of a single building block called monomers. Here we can actually see where that monomer, that single building block, is going to polymerize or go through polymerization and actually form different bonds that link together. And these linked togethers are going to link together multiple times to form, as you can see, a macromolecule of multiple monomers that have actually combined together. Now, of the different organic compounds, we'll start with carbohydrates. All carbohydrates consist of the following molecules: monosaccharide, disaccharide, and a polysaccharide. You'll notice that really they all have that suffix saccharide. Saccharide means sugar. It's a saccharide molecule, but it's going to just specify on the prefix as far as how many it has. Mono is going to be 1, di is going to be 2, and poly means many. So poly is going to be more complicated than a monosaccharide. Now, the larger carbohydrate, a polysaccharide, are comprised of many of the monosaccharides fused together. An example of this could be glucose, fructose, or ribose. Those are going to be a large amount of them. Now, glucose and fructose together are going to equal sucrose. You're looking at, in the body, is a lot of glucose molecules that are bound together. And glucose molecules that are bound together and stored in the body is called glycogen. Glycogen is going to be our stored carbohydrate in the body. Next, we actually have lipids. Lipids are a diverse group of molecules that can be hydrophobic and amphiphilic. So portions of the molecule are actually both hydrophobic and hydrophilic. So they can work in both aspects. Unlike other organic compounds, lipids do not form large macromolecules, nor are they built from monomers. Instead, lipids are basically long chains of carbon and hydrogen, a hydrocarbon chain that sometimes is linked with smaller molecules like glycerol. There are 3 types of lipids: fats, otherwise known as a triglyceride in the center, phospholipids that you see on the left-hand side, and steroids, which are formed of cholesterol on the right-hand side. Phospholipids are going to make up a large amount of the overall cell membrane layers. This class of lipid is largely responsible for the membranes around the cells, and phospholipids are similar to fats, but with only 2 fatty acid chains linked to a glycerol backbone, which also contains a phosphate group, and it's made up of phosphorus and oxygen. We'll talk a lot more about the overall layout of phospholipids and how they're involved in the cell membrane layers in the next lecture. The next one, triglycerides, are fats. Fats are a very efficient form of long-term energy storage in the body, but they can also function structurally. We use them in a lot of cases as different sorts of fat pads in the body to help against abrasion, shock absorption, and things like that. We also put them around a lot of breast tissue for females to make sure that it's actually padded there to deal with other abrasion. It can be used for insulation in the case of the greater omentum of the abdominal cavity to help maintain warmth in the digestive tract. Fats are a large lipid comprised of 3 fatty acid hydrocarbon chains and linked to a glycerol molecule, as can be seen above. This is why it is known as a triglyceride, tri meaning 3. The final class, steroid. Steroids are a class of lipids where the carbon skeleton forms 4 rings instead of elongated chains. The most basic steroid that we have in our body is cholesterol, which is also found in many animal cell membranes. Many animals also use this to make an important class of molecule called steroid hormones. Steroid hormones are going to be things like testosterone, estrogen, and our stress hormone, cortisol. The final overall classification that we'll talk about before we get to nucleic acids is proteins. Proteins are polymers of amino acid building blocks, and they serve a tremendous variety of functions, from structural to communication to enzymes and more. Here we can see a breakdown of the different amino acid protein groups. Now, proteins have a lot of different structures. Here we can actually see a lot of the different structures with respect to protein. It goes through a primary structure up top. Now, the primary structure, the protein, is made up of a unique sequence of amino acid monomers that make up the polypeptide chain. So it's a very simple amino acid sequence. From there, we get into the secondary structure. The secondary structure, in this case, the backbone of the amino acids, can create coils or folds of the polypeptide chain. It becomes what we call an alpha helix structure and folds over on itself. From there, we can get to a tertiary structure. The tertiary structure, the functional group of amino acids combine to one another, creating additional complex shapes on top of the helix or folds caused by the backbone. And the final structure we get to is a complex of protein molecules called a quantitary structure. The quantitary structure is the protein could consist of 2 or more polypeptide chains, and these can be linked together to form different shapes as well. So the more complex a protein gets, the more unrefined it is. As we actually break things down in our digestive tract, we eventually go backwards from a quantitary structure, which is going to be a complex of proteins together. We could do multiple quantitary structures together, and that might be like a piece of meat. And then as we eat it and go through digestion, we eventually go backwards and refine it down to an amino acid sequence. So effectively, proteins themselves are going to be amino acid chains that will fold on top of each other and become more complex and more numerous over time. The final organic compound that we talked about is, of course, nucleic acids. And a lot of people are familiar with nucleic acids in the form of DNA that we see at the bottom. DNA starts off initially as a nucleotide. It is a monomer. It consists of a phosphate group, a sugar, which is a ribose or dextroribose, and an organic base. From there, multiple nucleotides combine together in a single strand called RNA. This is a signaling or messenger that will thereby make a copy of itself and then combine to create the program that we see in the cell called DNA, which is a double-stranded nucleotide compound. And this is going to be information storage and retrieval is the primary function of this. So this is the basics of chemistry as well as body cavities for Lecture 2.